Assalamualaikum warahmatullahi wabarakatuh. I am Putra Santoso from Biology Department of Andalus University. With this opportunity, I would like to present of our research focusing on the beneficial effect of dietary fiber extracted from some local tuberous plants in Indonesia in counteracting diet-induced obesity. Consuming a high-fat diet excessively could promote the development of obesity, that's so-called diet-induced obesity. In our present study, we aim to investigate whether the dietary fiber extracted from the tubers of some local tuberous plants, including ganyong, mentawi taru, garut, and bengkuang, could counteract diet-induced obesity. We firstly extracted the fibers from the tubers of that plant. So we followed the procedures as previously described by Kamalasari, and we separated these fiber fractions and then dried it up in order to obtain the high concentrations of the fiber. And based on the protocols we applied for those extractions, we successfully obtained the fibers powder for each different kind of tuberous plant. Furthermore, we carried out the approximate analysis to characterize the fibers. So we obtained the data showing that the fibers extracted from the tuberous plant, including Canna, Colacacia, Maranta, and Passaceros, composed of low concentrations of proteins and also low concentrations of fat. Next, we also determine the concentrations of non-starch carbohydrates. And as shown by this data, that the fiber extracted from tubers of Canna, Colacacia, Maranta, and Passaceros contains higher concentrations of non-starch carbs, up to 90%. And we also determine the composition of the dietary fibers itself, whether it's composed of contains of water-insoluble and water-soluble dietary fibers. In this data shown that the insoluble dietary fibers is highest in Canna fibers as compared with others, and the lowest one in Colacacea's dietary fibers. And for the water-soluble dietary fibers, the fibers extracted from Passaceros or Bangkuang tubers contain high concentration of soluble dietary fibers, and the lowest one remains in Colacacia. Furthermore, we perform the experimental treatment in animal models, mice. So we divided animals into 6 different groups and treated with 6 different kind of diet paradigm. The first group were fed with low-fat diet, call it normal diet, and the second group fed with high-fat diet. And the other 4 groups fed with high-fat diet supplemented with 25% of dietary fibers extracted from Canna, from Colacacia, Maranta, and also from the Passaceros. Next, we determine the daily calorie intake. This data depicted that the supplementations of dietary fibers from Canna, Colacacia, Maranta, and Passaceros increase the daily calorie intakes in high-fat diet mice as compared with high-fat diet lot. And in particular, analysis to the calorie intake from fat that also reflected the daily calorie intake also revealed that the intake of calorie from fat was also higher in dietary fiber supplemented groups compared with high-fat diet plants. Next, we measure body weight for every 2 weeks during the treatment. And so it's shown here that in high-fat diet groups, the body weight rapidly increased starting from the second weeks of the treatment and became highly significant as compared with other groups, suggesting the obesity development in these mice. And in the control groups and also in dietary fiber supplemented groups, the body weight uh, was sustained in a normal increment. Shown here that the body weight increased, of course, but the trend of the increment was remained sustained in normal levels and became comparable with the control groups. Then we also calculated the body weight gains at the end of the experiments. So after each week of the treatments, we found that supplementations of dietary fiber extracted from these tuberous plants could significantly suppress high-fat diet-induced obesity. And the magnitude of suppression was highest in mice treated with fibers extracted from the kana. And at last, we also calculated the resistance of mice against high-fat diet-induced obesity under the different treatments. And we found that the resistance level was the lowest one in high-fat diet alone, of course. It is mean that mice become very sensitive to develop the obesity under high-fat diet treatment. But when we supplemented it with dietary fiber from Kana, Calacasia maranta, and Passocereus, then the resistance will be increased rapidly. And it is showing that the resistance of such mice against diet-induced obesity. In conclusions, our present study revealed that dietary fiber extracted from the tuberous plants, including Kana or Ganyo, and Colocasia, Menthawi taro, and Garut or Maranta, and also Bengkuang, Passacereus, could effectively prevent the development of diet-induced obesity in mice models. That also plausibly reflects the same response when it applied in humans in the future, of course. And among the four species of this tuberous plant that commonly found in Indonesia, we recommended that Ganyong or Canaidules as the most effective one in combating the diet-induced obesity in uh, high-fat diet treated. So we would like to express our gratitude to Indonesian Ministry of Education, Culture, Research and Technology for supporting our research. Thank you very much.